Okay, so now we start with the topic of apoptosis. Apoptosis is referred to as programmed cell death. Now this process is genetically controlled or controlled by the genes. So it can be both physiological and pathological. Physiological examples include like embryogenesis or menstrual cycle or some infection, whereas pathological changes which are cellular phenomenons, like there is an example of or basically a high yield point being Councilman bodies seen in viral hepatitis. So these Councilman bodies are formed due to the process of apoptosis, which is only seen in viral hepatitis. So if someone is having viral hepatitis and you see Councilman bodies, there will be the process of apoptosis going on in that patient. So now talking about the genes associated with apoptosis. Now the pro-apoptotic gene or the genes which promote apoptosis are going to be p53 gene, which is a very important pro-apoptotic gene, the BAC gene and the BAX gene. Now the anti-apoptotic or the genes which don't promote apoptosis are Bcl2 or genes like Bcl-XL. Now if we talk about detailed mechanism, now there are two types of mechanisms which we can see in apoptosis, one being the intrinsic pathway and another being the extrinsic pathway. So talking about the intrinsic pathway, we emphasize on the cellular structure first. So in the cell, you have the mitochondria and the mitochondria has cytochrome C inside it. Now the cytochrome C has to stay inside the mitochondria. If the cytochrome C goes out of the mitochondria into the cytoplasm, that it can promote apoptosis. So there is Bcl2 gene, which is an anti-apoptotic gene which prevents the cytochrome C from going out of the mitochondria. So when there is deficiency of growth factor or growth hormone, what happens is the Bcl2 stops functioning properly. So if the Bcl2 stops functioning properly, it is obvious that the cytochrome C will leak out into the cytoplasm. Thus the cytochrome C is released into the cytoplasm and it stimulates the caspases. So what happens if the caspases get stimulated? So if the caspases get stimulated, it is responsible for the stimulation of endonucleases and proteases. Now, what are endonucleases? What are proteases? Basically, we know that endonuclease destroys the nuclear materials and protease destroys the proteins. Thus, if there is no nuclear material and no protein inside the cell, so how can the cell live? Thus, the cell is obviously going to undergo cell death. So, this programmed cell death is known as apoptosis and the pathway is intrinsic pathway. Now, shedding some light on the extrinsic pathway. Now, what happens in the extrinsic pathway? If there is severe damage occurred to a cell, it causes the release of TNF alpha and Fas ligand. Now, what happens after that is they are attached to their receptors and activate the receptors. So once the receptors are activated, it is responsible for the stimulation of caspases. So the thing which we see common in the intrinsic pathway as well as the extrinsic pathway is that the caspases are stimulated by somehow or the other, which is the cause for apoptosis. So once the caspase gets stimulated, apoptosis is going to happen. Now there is an important point which I am going to talk about is the most characteristic finding of apoptosis is going to be chromatin condensation. So if you see chromatin condensation, you can surely say that the cell has undergone apoptosis. So the next thing I'm going to talk about is DNA gel electrophoresis and what we can see as a result. So if there is gel electrophoresis done of an apoptotic cell, we can see a step ladder pattern. Now there is a slight confusion which happens when we give the exam. So if there is a question saying that step ladder pattern found on fever or step ladder fever, we should not write apoptosis because that is associated with typhoid or enteric fever. So if a patient has undergone typhoid or had enteric fever, we can see a stepladder pattern on the fever or the stepladder fever. Now the stain which is used for the diagnosis of apoptosis is going to be annexin 5. This is a very important point to remember. Now comparing apoptosis with necrosis, apoptosis undergoes no cell membrane damage, whereas in necrosis there is visible cell membrane damage. In apoptosis, no leakage of intracellular enzyme is seen, whereas in necrosis there is leakage of intracellular enzymes. And in apoptosis there is obviously no inflammation, whereas in necrosis there is visible inflammation. Now the next thing which I'm going to talk about is the most important pro-apoptotic gene, which is the p53 gene. Now this gene is also known as the guardian of the genome or the molecular policeman of the cell, because it is like a friend to the cell. I'll explain why. Because the p53 gene helps in the repair of DNA. Now the few points to remember about the p53 gene is that if there is repair of DNA and the repair is unsuccessful, so it will increase the Bax gene activity, which leads to apoptosis, because Bax gene is a pro-apoptotic gene. Now the stage of cycle where the replication will arrest, you have to remember this is G1S phase. This is a very important paper 1 question. And another question you need to remember that the chromosomal location of the p53 gene, which is going to be 17p, and if there is p53 gene mutation, it will generally lead to malignancy. So what does p53 gene do? p53 gene helps in the repair of DNA. So if the DNA is repaired, then it's well and good, and if it's not repaired, the p53 gene induces apoptosis. So if there is mutation or problems with the p53 gene and the cells cannot undergo apoptosis, so the cell with the mutated DNA is going to persist, which will lead to malignancy. Now another point I want to mention is that these all are caspase dependent programmed cell death, which is generally called apoptosis. Now if the question asks that what is the caspase independent programmed cell death, it will be necroptosis. Now the important points to remember: number 1 is the caspase dependent programmed cell death, which is apoptosis. Second one is the pro-apoptotic gene, which is the p53 gene, anti-apoptotic gene, which is the Bcl2 gene. Fast ligand is also known as CD95. There are some questions where the fast ligand is not mentioned, they mention CD95, so be careful about this. So if they say that extrinsic pathway is related to which, so if there is no fast ligand mentioned and there is CD95 mentioned, so CD95 will be the probable answer. So the next one is the step ladder pattern observed on gel electrophoresis will be an example of apoptosis, where the DNA gel electrophoresis shows a step ladder pattern. Stain used will be annexin 5. We also have to remember the cytochrome C stimulate caspases. We know that when the cytochrome C is released into the cytoplasm, it promotes the stimulation of caspases. Now the most important organelle involved in apoptosis will be mitochondria. Stage where the replication arrests is G1S phase. And lastly, the chromosomal location of p53 gene is 17p.